Transcription and translation. In other words, protein synthesis. So this is how you make proteins. And so DNA being made to RNA is called transcription. Then you have to snip some of the pieces of RNA out and glue them back together. And when it goes out to the protein, when it goes out to the ribosome to make a protein, that's called translation. So let's talk about that a little bit. So protein synthesis, again, synthesis means to make. So making proteins. So protein synthesis is the production or synthesis of proteins. And another word for protein is polypeptide. So polypeptide chains or proteins. There are two phases to this, transcription and translation. So let's talk about that a little bit. So DNA makes RNA. RNA then gets modified. All of that happens in the nucleus because in order to make your RNA, you do that right from DNA, and the DNA is stuck in the nucleus, so this whole process happens in the nucleus. And then you send this out of the nucleus into the cytoplasm on a ribosome, and you make the protein. So this process is called transcription, and this process is called translation. Amino acids gathered by tRNA and brought to ribosomes, that's what happens here. So transcription. What does transcribe usually mean? So I used to work in a college where people learned how to be medical transcriptionists. They would do what I'm going to show you here. So here's the doctor, and she's speaking into a recorder. She's doing all of the notes from the day. And here's a medical transcriptionist, and she's typing up the doctor's notes. So that's what transcription means. It's in the same language. So both are, in this case, maybe English. Both are in the same language. So transcription is the process of copying a sequence of one strand of DNA, which is the template strand, into messenger RNA. This is really the same language, so we're calling it transcription because you're just changing it from a series of A, T, C's, and G's to the ones that complement, U, A, G, C. So you're going from DNA to RNA. But it's really the same language because those are all nucleotide bases. So they're both in the language of nucleotides. So that's the way that you can remember that transcription is DNA to RNA. This requires an enzyme. You're making RNA, so the enzyme is called RNA polymerase. So you can tell it's an enzyme because it ends in ase. You know what a polymer is. We studied that back in September. So monomers are the little guys. You string them together and you make a polymer. RNA polymerase is an enzyme that makes a polymer of RNA. So that one's really well named. So for example, here's DNA. This one is the template strand. So this separates out and you use that enzyme and you match these up. So T goes with A goes with U, C goes with G, G goes with C, G goes with C. And if you pause this and you look along the whole thing, you'll be able to see that this is the complementary strand to this. So from DNA to RNA, that thing, you can call it mRNA synthesis, or you can call it transcription. Question, what would be the complementary RNA strand for the following DNA sequence? So it'll start 3', G goes with C, C goes with G, G goes with C, T goes with A goes with U, because it's RNA, T goes with A, and G goes with C. If you're going to mess up here, it's going to be along here somewhere. So sometimes students will just write T to U by accident, but try to remember T goes with A. You can say A to T, and then say, oops, it's RNA, it must be a U. Okay, so during transcription, RNA polymerase binds to DNA, and you can see here, this blue part is the DNA, and it's unwounded. It's opening it up. So it unwinds or separates the DNA strands. And this green part is the new RNA that's being made. This whole orange thing is the RNA polymerase. So this is the enzyme that makes the RNA. So here's the RNA being made. So you unwind it, you separate it out, you put the strand that you don't need up here. Here's the template strand, and then you put the DNA back together. RNA polymerase then uses one strand of DNA as a template, to assemble the nucleotides into RNA. So if this were a G, then the RNA would be made here and there would be a C to match it. Promoters, that's this part. Promoters are regions of DNA that allow RNA polymerase to begin the transcription of RNA. So RNA can't just go into the middle of a gene and start translating it. The RNA polymerase, that enzyme, has to recognize this area. It's called a promoter. And there's a TATA box at the beginning, TATAAA. And then after that, there's some more DNA, and then there's the start point, and that's where the RNA will start reading. So it's called a TATA box at the beginning. So here's the RNA polymerase, and so it attaches to this whole thing. This is that TATA box that we were talking about before. Here are some transcription factors that attach as well. And then here you can see it's unwinding, and it's going to start reading the DNA right from here and making the RNA right at that spot, not at the earlier spot. Terminal signal, this is a specific base sequence that acts as signals to stop transcription. So you have hopefully I have a picture here. Good. So you have the whole DNA, the RNA polymerase is going along and reading it. At this point, this is the stop signal. And so that's where it just falls off. And these factors just separate out. And here's your mRNA all done. And then the next thing you do is process the mRNA. So after the DNA is transcribed into RNA, editing must be done to the nucleotide chain to make the RNA functional. So it's kind of crazy, but the RNA, let's say this is the messenger RNA, this part might be not useful, and this part might be not useful, and that part might not be useful, and maybe that part might not be useful. So this section, and this section, and this section, and that section will be put together into this smaller strand, and that will be the functional RNA that will go out to the ribosome. Introns, these are non-functional segments of DNA that are snipped out of the chain. So here's the original chain, here's the DNA, the template strand. Here's the RNA that's made, and let's see which color's which. So these are the intron. No, these are the introns that are cut out and cut. So these pieces, the yellow ones, are the introns. That stands for intervening sequences, the ones that aren't used. So that's cut out, that one, that one, that one. All these yellow ones are cut out, and the blue ones are, or green, sorry, the green ones are spliced together. So these might have some function, but they don't function in this gene. They might have some type of regulatory function, but they don't function to be translated into a protein. So introns are called intervening sequences. They're in between. Exons, these are the segments of DNA that code for proteins, and they are rejoined by the enzyme ligase again. So you heard of ligase before when we connected Okazaki fragments. Ligase is an enzyme now used to connect the RNA together. So these are the exons, and these are the introns. A guanine triphosphate cap is added to the 5' end. And so here's the 5' end with the cap on it. And then you get a poly A tail at the other end. Poly A tail is added to the 3' end of the RNA so that you have this so that it can leave the nucleus the right way and be processed by the ribosome the right way. You don't have to memorize what's on the beginning and the end, but I want you to get the idea that this whole thing is processed. So you cut out the stuff you don't need, splice together the good stuff, and then put a couple of caps on them, a cap and a tail. So the newly processed mRNA can then leave the nucleus. And so here's the cell. This thing's the nucleus, that's a nuclear pore, and here comes the mRNA out, and it will find a ribosome and start making proteins next. So the mRNA leaves the nucleus through its pores and goes to the ribosomes out in the cytoplasm. I know, isn't science cool, Shrek? Okay, translation. So translation is a word that you probably know a lot better. To translate something is to change it from one language to another. So I took 3 years of Latin, and it's scary because I think I remember about 3 or 4 words. And I took a year of Spanish. Let's see, that means Spanish, and that means talk, I think. So someone's gonna fix my awful Spanish. Um, do you speak Spanish? I don't know. They speak Spanish, maybe something like that. Anyway, I don't know what it says, but a lot of you do. So to translate something is to change it from one language to another. So I can help you translate into the language of amino acids, though. So what does translation usually mean? Changing from one language to another language. So translation is the process of decoding mRNA into a polypeptide chain. You're going from nucleotides into amino acids. Totally different. I can do the DNA to RNA thing without a special code book. DNA to RNA, RNA to proteins. That's the way it works, right? This part's easy. If you give me, or any of my freshmen, a list of DNA, most of you can change it into RNA. If it's G, G, C, A, T, T, most of you could tell me G goes with C, G goes with C, C goes with G, A goes with U, T goes with A. Most of you can do that. That's called transcription. That part's the easy part. Now, what does this code for? And what does this code for? What amino acids? Heck, I don't know. I'll have to look it up. That's a translation. I think that's a harder thing to come up with. And so we use a cheat sheet for that because I'm not going to make you memorize that for goodness sakes. But does it make sense that it's a new language? So that means translation. So you do need to know that. So we are moving from the language of nucleotides to the language of amino acids. So you're moving from this, the language of nucleotides, UCCAA, to the language of amino acids, that one, then that one, then that one. Ribosomes read messenger RNA 3 bases, or 1 codon at a time, to make the proteins. And so if you look at this, here's your DNA, and here's the mRNA made from it. That's called transcription, G to C, C to G, C to G, A to U, T to A, etc., etc. You bring that out into the cytoplasm, to a ribosome, and you read 3 letters at a time. So that codon codes for arginine. That's the amino acid right here that's coded for by this code. This code, UAG, when you go to the ribosome, there'll be a tRNA that will always take over tyrosine and match up with this one. It'll have an anticodon that will match this. It would be AUC, and it will always bring over tyrosine. And then this one, ACC, that codon will always get matched up with tryptophan. So these are amino acids that are going to be put together to make a polypeptide. So this is translation. You're going from the languages of C's, G's, U's, and A's to the language of amino acids. Which amino acid comes first? Which one comes second? Which one comes third? Okay, so this is where I'm going to kind of get into some of the details. I think I'm going to stop here because it's going to get, I think it'd be a really long video if I don't. So I'll pick up here next time.